So welcome guys once again. On in this video we are talking about the biochemistry, some short notes or we can say some terminology which will be interconnected to each other, and overall motive of this video is to revise some topic or to touch some topic that will be help us to clear some exam or identify, evaluate ourselves, right? So first of all we go to the carbohydrate, which is one of the main important chapter in the biochemistry. So carbohydrate, so they are classified into monosaccharide, disaccharide and polysaccharide. Monosaccharide mean simple single type of carbohydrate like a glucose, fructose, galactose and ribose. So these are the common example. In case of disaccharide, when two monosaccharide combine, it become a disaccharide. Let's suppose there is sucrose, lactose, maltose. So these are all the disaccharide. So when we combine glucose and fructose, then become a sucrose. Sucrose is a sugar cane. So sucrose is a cane sugar, right? Another we have a lactose, which is a milk sugar, right? Lactose is commonly seen in milk. So it is made up of two type of glucose and galactose, become a lactose. Another one is maltose, mainly in our cereals and some fruit also contain this maltose. Wheat, all these things contain maltose, which is made of two molecule of glucose, right? Glucose and glucose, maltose. So these are also important thing, come in exam in different way, right? When more than 10 monosaccharide combine each other, then it become a polysaccharide, right? Very important thing, more than 10 monosaccharide when combined, it become a polysaccharide. Like a starch, which is a storage form in case of plant, right? Starch is a storage form in plant, while in case of glycogen in animals or in human being also, okay? So in case of human being, our storage form of carbohydrate is glycogen. In case of plant, it will a starch, which is a very important. Another some polysaccharide also play help in structural role, okay? It maintain the structure of our plants, such as cellulose and chitin, right? So in case of cellulose, there is a very good thing that it can't be digest, right? So cellulose can't be digest, it will be removed from the body as it is, right? Now we talk about the metabolism of our carbohydrate. So it is a very vast topic, but we are touching some important thing, so it will help you or remember some points for your upcoming exam, right? Kind of revision. So there is a word glycolysis, which means breakdown of glucose into pyruvate or lactate, right? So in this condition, glucose breakdown to pyruvate, or in the anaerobic condition, it goes into the lactate condition, right? So these are two types, anaerobic and anaerobic or aerobic. In case of aerobic, pyruvate is the end product. In case of anaerobic, lactate is the end product, right? Another we have a gluconeogenesis. As the word suggests, gluco means there is a formation of glucose, but from the new sources, right? Genesis means formation. So if we degrade the word in a simple way, so we can identify the term, right? So you can see the formation of glucose from non-carbohydrate source like glycine, amino acid. If the glucose made from this type of product, then it come a term gluconeogenesis. Another one is a glycogenolysis. Glycogen, so it means, and lysis, breakdown, right? Breakdown of glycogen into glucose, it become a glycogenolysis. When the body need immediate glucose, right? In case of low energy, body need a glucose. So at that time, the process glycogenolysis is activate, and it will help to provide us glucose, right? Another one we have a pentose phosphate pathway, very important pentose phosphate pathway. It secrete a NADPH, or we can say during this cycle we take this NADPH, and this is very important for our formation of different kind of product in the metabolism, right? So it is very important. Then we have also some important tests to identify. One of the simplest tests is Bending test. It detects reducing sugar, right? And color change from blue to red, right? So in case of Bending test, it mainly detects the reducing sugar, right? So all are the reducing sugar except from the sucrose. Sucrose is non-reducing sugar. So here is another point, sucrose is non-reducing sugar, right? Another one, we have a trehalose, which is also a non-reducing sugar. So we can't detect through the Benedict test. For that we do some other test, like a enzymatic method. So that is a different procedure, or we use some kinetic method also, right? So this is a we discuss later on different test. Another one, we have a Barford test. Barford test, different monosaccharide from disaccharide. So basically differentiate monosaccharide or disaccharide, right? Iodine test, it is a common thing. Usually you can also see in the NCRT book in 8th 9th. So we can detect starch through the iodine test. It become blue to black color, right? If there is starch, so it will convert blue to black with the help of iodine test. You can also test, you do this with some tincture iodine solution, which usually you find in your hospitals. So put it on the potato, and if it turn into black, it means it contains starch. Obviously potato have a starch. Now another thing important is protein and amino acid. So in case of amino acid, we have two type of amino acid. One is essential amino acid, another is non-essential. Essential, it means it is not synthesized by body, we have to take from the outsource. So they are this is a mnemonic for that, you can remember AVHIL MPTT. So alanine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, tryptophan, and wait, threonine and tryptophan. So these are the 10 essential amino acid, right? You can also learn this terminology, and this is a technique you can remember. Although I'm not saying I'm very perfect in all this thing, we have to I have also prepared something, and maybe during I'm teaching you, and in this case also I forget something, but this is the human nature, right? Because I'm not very detailing about this topic, but so we go to another, like if we said about the protein, what is the protein function? So it role play in structure, transport, enzyme, and hormone level. So in case of structure, collagen and keratin both are the important thing of our body, right? Keratin hair and nail, and collagen all over the body we can find. Transport, so when the protein combine with some other molecule, so it transport, like in case of hemoglobin, right? Here the globin is the protein and hem is a product. So it help transportation of, right? Albumin, it also transport one place to another place, maintain the balance in the body. Protein also act as some enzyme, like a catalyst for biochemical reaction. So in a biochemical reaction, if we want to fast the process, so we need a catalyst. So protein also act like an enzyme. Or some protein also act as a hormone, like insulin, glucagon. Insulin reduce the sugar, while glucagon increase the sugar inside the body. So they are very important role in the body. When someone have an insulin deficiency, so we need to put from the outside, and you can imagine the cost. That's why the body is very important, or the beautiful gift of nature. So protect it. Now we talk about the protein metabolism. So protein metabolism mainly two terms, deamination and transamination. Removal of amino group. So basically protein have amino group. If we remove that from the protein part, so the process or the term known as a deamination. Transamination is when we not remove but we transfer from one amino group to another amino group, that is common the transamination. So in case of protein, urea is the end product, but urea is very toxic for our body, so we have to remove it, right? Sorry, ammonia, which is very toxic, so we have to remove it. It will be removed with the help of this urea cycle. excretion of nitrogen waste, right? So urea and this is also known as a Krebs cycle. Sometimes it is also known as Krebs cycle. So for the exam point of view, very very important thing. Now there are different tests for the protein, like bio test for peptide bond, violet color we can see. Nin hydrogen test is also important test. It measure particularly some amino acid. Overall we have to measure some protein or peptide, then we go for biorate. But for specific amino acid we can go for the nin hydrogen test, purpose of the test if it is positive. Then we have a xanthoprotic test. Basically it detects the aromatic amino acid. So when we talk about amino acid, it is very vast chapter. We have a different type of sulfur containing aromatic amino acid and some other type of amino acid. But if we detect the aromatic amino acid, it will, the color represented as a yellow color, right, for the positive one. Now another one we have go to lipid. Type of lipid: simple lipid, complex lipid, derived lipid. So in case of simple lipid, as the name suggests, simple, it contains fat and oils. Complex lipid made of two simple lipids, phospholipid and glycolipid. Phospholipid, it means it contains phosphorus group addition to the lipid. In case of glycolipid, addition of glycogen to the lipid. Derived lipid, so basically they are derived from the above two type of lipid, simple or complex, like a steroid and fatty acid. Now go to the lipid metabolism. So beta oxidation is very important in case of beta oxidation in lipid metabolism. Fatty acid breakdown for energy. So basically it breaks down the fatty acid through this beta oxidation pathway. Another one is ketogenesis, formation of ketone body in the fasting starvation. This is very important as how the ketone body form. So they are formed during the fasting and starvation condition, right? If we see the number of amount of ketone bodies, so it means the patient may be in fasting stage and starvation, and it will lead to some body disorder, right? Another we have a cholesterol synthesis. So in case of lipid, we have also important thing, the precursor for steroid hormone. So cholesterol synthesis is very very important for overall the body cell membrane, cell and other parts of the body. We need a cholesterol for equal balance for the body. Now another we have a lipid profile test. So lipid contain total cholesterol, LDL, HDL, TG, triglyceride. Here is the normal value, less than 200 mg per deciliter. In case of bad cholesterol, it is less than 100 mg per deciliter. But HDL is a good cholesterol, which is more than 40 mg per deciliter, and TG is 150 mg per deciliter. So you can go from low to up and up to more up, right, in this condition. So LDL is very dangerous for the body. Note, LDL is very dangerous for the body because it is a bad cholesterol. It transport all the cholesterol and blockage our heart system. But HDL is good cholesterol, it remove all the thing, right? Another we have a topic, and that is enzymes. Enzymes, very very important. So we have different properties for the enzyme, like a catalyst. First and the main property of enzyme is it act as a catalyst. In a simple form, catalyst is nothing but to increase the specific reaction, right? So you can see here the written speed of the biochemical reaction. If any of the biochemical reaction is speed up, it is due to catalyst, like, right? Another property is enzyme is very very specific, like a lock and key system. So glucose enzyme will only act on glycogen, right? So it is very specific. We can't say the protein enzyme act on the glucose or another one. Yeah, there is some chances that known as a miscellaneous, but main thing is that it is not as usual, right? So we need some cofactor also for the accuracy of this speed of enzyme. When we have to perform some reaction, so it needs some cofactor also, like a magnesium and zinc is very very important for this cofactor. Another we have need some coenzyme such as vitamin derivatives. So these are some other thing. So enzyme also act as an inhibitor, right? So competitive inhibitor compete with the substrate. So when we have to analyze the test, two type of thing. So maybe one component is our original thing and one is just a duplicate. So it will help to identify that the competition is more or less, right? For some tests, it is very important feature for the enzyme if we see this competitive inhibition. Another is non-competitive inhibition, bind to allosteric site. Allosteric site, so what is allosteric site? So enzyme have a area where the another, like a key and lock system. So enzyme have some space where another thing has to come and attach that area. But sometime enzyme also have some another space. So this is known as allosteric site, where some if some another particle came or attached. But if so, this is a competition, right? If enzyme have only one space like this, so two particles have to compete each other, right, to bind here. But some enzyme also have some allosteric site, we can say the non-binding site, but the other side. So at this site another foreign particle may be attached, so it will act as a non-competitive inhibitor. Uncompetitive inhibition, bind only to enzyme substrate complex. This is something else. Now we go for the clinical enzyme test. So in case of enzyme, we have a liver function ALT, alanine transferase, and then aspartate transferase, ALP alkaline phosphatase. So these are the enzyme used in many liver function tests. Also we have heart in case of cardiac function, or we can say the used in myocardial infarction. So we have enzyme CKMB and troponin. CKMB also divided into CKMB 1, 2, 3 based on brain, body or bone level, right? Pancreatitis also a kind of enzyme such as amylase lipase. It will help us detect whether our pancreas is fine or not. Maybe it contains some disease, pancreatitis. So at that time amylase lipase both are increased. In case of myocardial infarction, CKMB, troponin. Tropi is very important test, right? Myoglobin also. Some other there are so many tests, but in case of specifically enzyme case, CKMB is very important. And in case of liver, ALT, AST, ALP, these are for the liver. Another we have a topic vitamin. So we have a two type of vitamin, water soluble and fat soluble. So you can also remind the name because these come under exam. B1 thiamine, B2 riboflavin, B3 niacin, B6 pyridoxine, B12 cobalamin, right? From the B1 we have a disease beriberi. From B2 glottitis, so mainly the lip or around the lip we have some problem, right? It also help in metabolic maintain the metabolic state. B3 niacin, plagra. So it is also known as a triple D3 disease. Triple D disease, because you can see the patient having a diarrhea, dermatitis or dementia. So brain level problem, skin level problem, and the around our stomach, right? B6 very important for neurological symptoms. They come under neuropathy. B12 pernicious anemia. So in case of female, B12 is very important during the pregnancy. Vitamin B, vitamin C, common our scientific name is ascorbic acid, and it causes scurvy. There is cut around the corner of the leaf, our lips. Another we have a fat soluble vitamin. So ADEK, sometime it become a KIDA also, right? Short form KIDA. So vitamin A cause night blindness. Vitamin D rickettsia, osteomalacia rickettsia in children, and osteomalacia in adults. Vitamin E antioxidant, and it also important in hemolytic anemia, right? Vitamin K bleeding disorder. If this vitamin K is absent, so patient may see the bleeding disorder. 2, 7, 9, 10 is a vitamin K dependent factor. So if we have a deficiency of vitamin K, this factor 2, 7, 9 and 10, clotting factor. So this is very important, right? Please remind, this is all the important thing. Now come to another thing, which is the acid base balance. We come into the pH. So blood pH is normally 3.35 to 4.5. Acidosis, when the blood is below the blood pH is below the 7.35, Alkalosis means alkaline state when the pH is above the 7.45. Buffer system, we have a bicarbonate, major blood buffer है. So sometime question come in exam, what name the major blood buffer, so it is a bicarbonate buffer. Intracellular fluid buffer is phosphate buffer. We also have a protein buffer like a hemoglobin. It transport the blood but also act as a pH maintain as a balance, right? So albumin also play a role in case of balancing the pH, maintain the pH of the body. This hemoglobin, albumin. So you can see the bicarbonate, phosphate and protein buffer. And protein we have a two type of albumin and hemoglobin. Another one we have a metabolic metabolism and energy production. So these are also a very vast topic. So carbohydrate metabolism cause glycolysis, glycogenesis. We already discussed all these things. In case of lipid metabolism, beta oxidation, ketogenesis and cholesterol synthesis. Cholesterol is very important. It will also go toward skin. What is the important term? Toward skin, right? And protein metabolism, we have a term deamination, transamination and urocycle, which is on Krebs cycle, right? Now another thing is hormone and clinical biochemistry. Hormone and clinical biochemistry. It contains hormone, first of insulin, lower the blood glucose. So insulin mainly reduce the blood glucose level. Glucagon increase the blood glucose level. Cortisol stress hormone hai, and it also increase the glucose level. Thyroid hormone T3, T4 and TSH regulate or balance the metabolism, right? Adrenaline in the PUBG game also you see the adrenaline injection. So it mainly a life-saving thing, right? It increase the heart rate so that the body pump the blood all around the body and maybe patient get chance to survive. It is also known as flight or fight response, right? If you come, if the lion come in front of you, so the adrenaline level increase too high and you will to fight or flight. So both are the thing common, that is adrenaline hormone, right? Another one we have biochemical test. So these biochemical tests such as blood glucose test, right? Blood glucose test in case when we monitor diabetes condition, in case of diabetic condition. So there are so many things come under the blood glucose test. Fasting blood glucose level 70 to 100 mg per deciliter. In some case it may be 110. So based on lab to lab, we can identify through the Godput method, glucose oxidase peroxidase method. But hexokinase is very specific because it also examine the non-reducing sugar, right? Then we have a PP, post prandial blood sugar level. So we do this after 2 hours of meal. Normal is less than 140. We can do this with the Godput. Random blood sugar less than 140. Godput method or hexokinase, both we can use. HbA1c, very important. HbA1c, it is the tell us the 3-month sugar level. 3-month sugar level. We can do this with the HPLC or immunoassay. Normal range is less than 5.7. Pre-diabetic or we can say borderline 5.7 to 6.4. More than 6.5, it is a diabetic condition, right? Very important test if we have to clear cut whether the patient diabetes or non-diabetes. So based on this value we can identify. Another one, we have a oral glucose tolerance test. What is this? So we basically give glucose by orally to the patient, or we can also inject, right? So these are two another thing. So it is very important for gestational diabetes. In case of mother having a baby inside the womb, so at the time she having a diabetes or not, we can identify with the help of this OGTT. Very important. Less than 140 mg per deciliter normal, and in case of impaired 140 to 200, more than 200, it become a diabetic condition. Basically we check the how the body responds toward the glucose load if we directly give to the patient. Another one, we have C-peptide test. Normal is 0.5 to 2 nanogram per ml. Basically indicate insulin production. It will help to give us how the insulin is production inside the body. We can do this with the immunoassay method. Now let's go for the liver function test, LFT. In this we have a total bilirubin level, which is very important for jaundice, right? In case of number of RBC breakdown, patient lead to the jaundice condition, right? So 0.32 or sometimes 0.2 to 1.2 mg per deciliter is very normal range. Little changes in this range can create a huge problem, right? So there is a direct bilirubin and indirect bilirubin. The range is 0.0 to 0.3 mg per deciliter. Another we have a L-aligned aminotransferase, ALT, SGPT. L denote here to liver. So basically it tells us the liver condition. Normally 7.56 7 to 56 unit per liter. Basically liver cells के लिए. We also use method kinetic UV method, right? Another we have a aspartate aminotransferase, SGOT, liver, and also it tells us the heart disease, heart damage, 10 to 40 unit per liter. The range can be changed based on your lab to lab. Another we have alkaline phosphatase. Basically it help us the bile duct condition, right? If there is stone in the bile duct, so we can identify with the help of this. Also bone disease, very important. 44 to 147 unit per liter is the normal range. PNP, para-nitrophenyl phosphate method, we commonly use for identify this. Then another we have a GTT, gamma glutamyl transferase, 8 to 6 unit per liter. So mainly this test alcoholic liver disease. If the patient have a alcohol damaged liver due to alcohol, then this test must come positive, right? Gamma glutamyl substitute method. This is a method name. We have a total protein method, Biochemistry method, as we discussed previously also. The normal range is 6.4 to 8.3 gram per deciliter. It will tell us the liver state or how much the protein as a nutrition available in our body. Albumin is also a part of protein, 3.5 to 5 gram per deciliter. So it will help to evaluate our liver and kidney disease, because kidney have to filter all this heavy protein and liver may generate this, right? So we can identify this with the help of BCG, bromoglycerol green method, very important, remind. Sorry. Now we go to another topic, and that is renal function test. So for the kidney we have also different type of tests, such as serum creatinine. So the value of this creatinine is very important. Minute changes can lead to a serious condition. In case of men, 0.7 to 1.3 mg per deciliter. In case of women, 0.6 to 1.1 mg per deciliter. And it helps to evaluate our kidney function test. JAF reaction and creatine JAF reaction, you can hear the name for the test method. And we also have some enzymatic method. Blood urea nitrogen method, BUN, 6 to 20 mg per deciliter. It helped us to evaluate protein metabolism, kidney function test. Urease method is one of the important method in case of blood urea nitrogen. Another we have a urea. So in this case also we have a urease method, 10 to 40 mg per deciliter is normal unit. In case of serum uric acid, uricase method is very important. One is urease, and one is uric, as an uric for the uricase, right? In case of men, 3.4 to 7 mg per deciliter. In case of women, 2.4 to 6 mg per deciliter. Very important, the uric acid level increase in case of gout, right? Arthritis. In this condition we see the increase in our uric acid. It also helps us to evaluate kidney disease. In the renal function test, we also have some electrolyte, sodium potassium chloride bicarbonate. So with the help of ISC, selective electrode, through this method we can identify all these things. You can note the normal range. Now another we have a lipid profile test, which help us to evaluate cardiovascular problem, or we can say in the short form heart disease. Total cholesterol less than 200 mg per deciliter. In case of male and in case of female, it can be less than 230. So there is so many method. This method named CHOD PAP method, right? Another we have LDL bad cholesterol, HDL good cholesterol. So you can note this. I will also send this overall PDF in the Telegram channel. You can join it, which is also help to make more note on this also, right? Another, we have a TG, triglyceride. We have some cardiac biomarker also for the heart disease, troponin, IT, right? You can note this, the reading. It is very important for heart disease. Gold standard for our heart attack is TROP-T, which is very important. Another, we have a creatine kinase CKMB, which is very important for heart disease. Brain natriuretic peptide, it is also very important for the heart disease. You can note the normal range. D-dimer test also very important, less than 500 nanogram per ml normal reading. DVT, deep vein thrombosis, in case of we can see this type of elements like a D-dimer. We can identify this with the latest agglutination or ELISA. We also have a thyroid function test, TSH, thyroid stimulating hormone. You can note the normal value. We have also T3. All this maintain the metabolic, right? Metabolic state of body, right? Very important for metabolism. We have an anti-TPO, thyroid pulsar antibody, very important. Sometimes anti-TPO test done for the thyroid function evaluation. It also help to get us in autoimmune disease, right? Basically, the antibody is against the thyroid system. Normal so overall metabolism is disturbed due to this. So in that case, we do the anti-TPO test. We have another thing also, inflammatory marker, infection, autoimmune, like a C-reactive protein, less than 10 mg per deciliter, normal range more than 100 mg. In case of severe condition and inflammation, we can identify this with latex circulation test, immunoturbidity method. We have a ESR in case of infection. We can also see the increased level Westergen or Wester Weintraub. These are two type method. We can identify with that the ESR level. Prolactin, prolactinone, which is also increased in case of severe infection or sepsis. So I know this is not enough video, but I am trying to make some more video. So basically, and I am also time bound, that I not take more and more time for you. So this is very short and quick recap you can gain from this video. I know it is not enough, but let's go. How much I can do, I will do. So thank you, all of you.